What'd you have for breakfast? Did you have sugar? That's actually fine. It's not as bad as you might think. And while it's true that we do study sugar molecules in my laboratory here, it's not the same kind of sugar that we think about when we're eating food. It turns out there's a whole different world of sugar biology, and it's taking place as a coating on the surface of all of our cells. So you might not have known that your cells are coated with sugars. I didn't know that myself until I was an undergraduate taking a biology class. And this would put us back to 1980. And in that class, our professor had not much to say about sugars at all, other than they're a coating on the cell, which means our cells are similar to peanut M&Ms. And when I was an undergraduate, that was about as much as people knew about sugars. In fact, our professor told us that the function of these sugars is probably like the M&M, where it's a protective shell that would keep the cell from melting in your hand, right? Remember that one? People of a certain age, M&Ms melt in your, but not. That's right. And that was the end of that. I remember thinking, well, that doesn't sound very interesting. Is that really all there is to know about cell surface sugars? And that was all our professor could tell us. But again, that was Harvard University. What do you expect? It turns out it's much more complicated than that. So the coating of sugars on our cells is actually much more similar to the coating of our planet with vegetation. The sugars are complicated. They're long, branched chains of complex carbohydrates. They come in many different shapes and sizes. Some of them are tall trees, others are little shrubs, and some of them are wide open fields of grass. And they're moving, and they're swaying. And it turns out they are an information data set. Different types of cells have different patterns of sugars, and it means something. Fast forward to 2023, and now we know that those complex carbohydrates that coat the surface of our cells are actually communicating with the outside world. They are a language that cells use to communicate their status. And when I say status, what I mean is whether they're healthy cells or whether they're diseased cells. Now, I became interested in these cell surface sugars when I started to read about how the patterns change in the context of cancer. And in particular, there is a type of sugar. It's a chemical structure that's shown here. Biologists often describe this sugar with a pink diamond shape. That's a symbolic language that the sugar community uses. And this particular sugar goes by the name sialic acid. Now, don't confuse this sugar with other acids. When you hear the word acid, you might think of a corrosive substance, but this is not that kind of acid. This is just a sugar, sialic acid. And all of our normal healthy cells will have a certain pattern of sugars that have the sialic acid as a component. I like to think of that pattern on the healthy cell as being like a well-manicured garden. Nice and neat, nice and trim, nicely organized. But if you look at the sialic acid sugars in cancer cells in a tumor, you'll see a very different landscape. On the tumor cells, there's a lot more sialic acid, and it's no longer a well-manicured garden. It has overgrown into a tropical jungle. There's a forest of sialic acids on cancer cells. That observation was made more than half a century ago, but until about 10 years ago, the scientific community had no idea why this might happen. And this was a mystery that we study in my laboratory. It turns out the reason cells have so much sialic acid in the context of cancer has to do with our immune system. So let me tell you a little bit about your immune cells. Your immune cells are the white blood cells, which course around through your body, arm in arm with the red blood cells. Those white blood cells have a job, however, which is very important. They are like the military-industrial complex that is defending you from disease. They actually protect you from cancer most of the time. And the way they do that, as you can see in this picture, is the immune cells, which are the green balls, they snuggle up to all of your cells one at a time, patrolling your body, and they literally taste the surface of your cells. And if your cells taste good, that means they should be healthy cells, and the immune cells will move along to the next cell and test it. But if those immune cells can taste a bad taste, that tells them this is a cancer cell, we should destroy it. And most of the time, that works quite well, which is why most of the time we are free from cancer. But of course, sometimes we're not so fortunate. Now, let me show you what might be happening when the immune cell touches the cancer cell. The immune cell has proteins on the surface. We call them receptors. They're like a tongue that's doing the tasting. And a cancer cell will have signals on its surface that are indicators. Hey, something's wrong with me. You should get rid of me right now. And that's what the tasting looks like. A receptor on the immune cell sampling a molecule that's on the cancer cell, and it tastes bad. This was a real huge breakthrough in the practice of oncology, because understanding the important role the immune system can play in protecting us from cancer is what led to the development of a new generation of cancer medicines that we call immune therapies. How many people, in passing, might have heard about cancer immune therapies? And maybe some of you have some direct experience with these medicines or know someone who does. They have absolutely transformed the practice of oncology, to the point where we now know that a good immune therapy will help an immune cell launch a bomb so that it can destroy the underlying cancer cell. And these medicines that cause immune cell bomb launching really took the news around 10 years ago. In fact, the breakthrough of cancer immune therapy was named Breakthrough of the Year by Science magazine back in 2013. Nature magazine, this is a very important scientific journal, also published on cancer immune therapy as a transformational, generational discovery. Even in the supermarket checkout line, you could read Newsweek or Time magazine and hear about the revolution of cancer immune therapies. And famous people were having their cancers cured. Do you remember when Jimmy Carter, former president, was diagnosed with advanced metastatic melanoma that had gone to his brain? That was a diagnosis that was 100% fatal before the advent of immune therapies. He was very lucky. He was one of the first wave of patients treated with an immune therapy, and he was very fortunate because it actually cured him. And using the word cure in the same sentence with advanced metastatic melanoma was completely unprecedented. Now, unfortunately, for every patient like Jimmy Carter who benefits from immune therapies and even has a cure of their disease, there'll be another 1 or 2 patients that don't. So even with all of these breakthroughs, most cancer patients still don't benefit from immune therapies that are now available. And the big question has been why? Why did that medicine work for Jimmy Carter, but didn't work for 2 other patients that looked on their face to be very similar to him? And this is where the sugars come in. Because what we discovered is that some cancer cells might have a bad taste for an immune cell, but if they can figure out how to coat themselves with that sugar, sialic acid, well, there's another molecule on the immune cells that when they taste the sialic acids, they deliver a signal to that immune cell that says, hey, I think this tastes pretty good, actually. Let's just walk away. So if a cancer cell can grow a jungle of sialic acids, it has an advantage that will be selected for just by evolution. And that is why so many cancers have so much sialic acid. That allowed them to trick the immune system, survive another day, grow, and spread, and cause disease. Now, once we had figured this out, we had an idea for how to make a new kind of immune therapy that goes right after the sialic acid sugars. And remember, I like to use the metaphors of vegetation for these complex cell surface sugars. And so we envisioned a medicine that would literally function like a molecular-sized lawnmower. And we had the vision of parking this lawnmower on the surface of a cancer cell, cranking up the engine, and just let that lawnmower mow the lawn. Because if we can take the sialic acids off the cancer cells, now they're going to taste pretty bad to the immune system, and our immune cells can do their job. And the great thing is when you're on the faculty here at Stanford, working with brilliant students, such as many of you, you not only can have ideas in the laboratory and do the research in the laboratory, you can spin out your discoveries into companies. And that is a popular route for many of our students who would like to take their scientific discoveries from the laboratory into the clinic to try to improve the lives of patients. So students and postdocs from my lab did just that, and we formed a company that we named Palion Pharmaceuticals. Palion has now made a lawnmower medicine candidate, which actually has the shape that I'm showing here. And that medicine is in a phase 2 clinical trial in a number of different sites around the country, but most excitingly here at Stanford University. And I'm very proud of my students for having achieved that. So, yeah, thank you. But I just wanted to leave you with this final picture once again. I hope this isn't exactly what you had for breakfast, but if you did, that's fine. Because your cells are like peanut M&Ms. They do have a sugar coating, and it's much more important than protecting those M&Ms from melting in your hands. So with that, I hope you enjoy all of the events of the weekend, and thank you very much.